So thanks everybody for joining this seminar. It's a really exciting series, and I think the network's been really interesting. I've learned a lot from participating. So let's hope that we have more of these kind of things. One of the things I'm strongly advocate is that we try and build more networks of people interested in especially diagnostics innovation. So just as a brief introduction, I originally did a PhD in immunology and spent a lot of time peering at dendritic cells and doing fundamental science. And then really the sort of middle part of my research career moved into applied um, research, starting off looking at vaccines, which is kind of a fairly logical progression from immunology. So if immunology is understanding the immune system, vaccines is manipulating the immune systems, and then got increasingly entirely absorbed in diagnostic technology, and especially this area of low-cost microfluidics, which I'm talking about today, and also became more and more active in innovation alongside research. And for example, I've been involved in startup companies, and a lot of that work was done at the University of Reading. So for about 13 years, I was in the School of Pharmacy at the University of Reading. And I'm really excited now to be, I guess, fully fledged engineer, if you like, in the sense that I'm employed by an engineering department and I teach engineering at the University of Southampton. And I'm in this very exciting group called Digital Health and Biomedical Technology Group within the School of Electronics and Computer Science. And as you'll see from my talk, I'm sort of increasingly looking to the future at how digital health is going to really impact diagnostic device development, and I'm going to try and raise some questions and opinions. But overall, my primary research can be summarized with these two words: diagnostics innovation. So in this presentation, in this webinar, um, I'm going to try and identify a couple of examples of grand challenges, if you like, in diagnostics. Both of these with question marks, because I think there's a lot of uncertainty about future directions. These are important trends, but not always as simple as meets the AI. The two big challenges I'll talk about are the challenge of near patient testing, and also, everybody wants their tests to be done near the patient rather than samples sent off to a lab. And then the other thing I'm going to talk quite a bit more about is this idea of integration and multiplexing, where we try and make multiple measurements all in one go. And we've done a little bit of work on that ourselves, and I think there's some interesting future trends in that direction, some opportunities, but also challenges. To illustrate these challenges, I'm going to talk about a technology platform that I've been involved in co-developing for probably over 15 years now, which is a technology platform for miniaturization. It's a little bit of unique, but as you're probably all aware, there's a lot of work on miniaturization and microfluidics across the world. So we're just one example of that, but I'm going to give an example of a point of care cell measurement that we've been working on to really address this question of point of care and how miniaturization allows near patient testing. I'm then going to talk a little bit about multiplex immunoassays, specifically in blood testing, and then also talk about seeing whether we can learn from both of these first two examples to measure blood cell function directly in blood. And through these sort of technical examples, I'm going to lay out lots of my opinions and try and share some of my experience and try and raise some questions about things which are challenging for the future. And we're always taught in innovation that every challenge is an opportunity. So I'm quite a positive person. So although I might highlight a lot of challenges and difficulties, hopefully these are all solvable. And I hope this all brings us back into the purpose of the Future Blood Testing Network by picking examples and thinking about big challenges. I think we can only really overcome some of these challenges if we share experience and work together. For example, maybe we need to do work with policymakers or regulators, or drive some shared research where we tackle some of these problems. So first challenge, it is now possible. Lots of people have shown that it's possible to miniaturize analytical bioassays. So what was once traditionally laboratory medicine and laboratory biochemical tests can now be, first of all, made smaller, and secondly, made automatic so that you don't need as much of an expert user to make them work. And this miniaturization process allows us to move testing to the point of care. And really, it's a kind of open question. I still don't know entirely whether this is correct, although my research is very much focused on making point of care prototype devices. I still think there's some challenges before we can have everybody just, using a magic, health test, Star Trek style, where you buzz the patient and you get a magical treatment answer. So I think there's a lot of compelling reasons why we want point of care, but it's not as easy as it looks. Um, in a way, the technology trends or the technology need can be summarized in this way. We have, on the one hand, low cost, widely available near patient tests such as urine dipsticks and lateral flow tests, which we all became familiar with in the pandemic for COVID home testing. And then on the other hand, we have incredibly high performance analytical instruments in laboratories. So microbiology labs in hospitals all now have mass spec MALDI capability, which is really astonishing. And the idea is that by improving analytical sensitivity of a diagnostic test, we can also then have a knock-on effect of improving the diagnostic sensitivity and the diagnostic performance. So that's the kind of trade-off at the moment. You can have cheap or you can have quality, but you can't have both. And what we're really driving towards through miniaturization and automation is this right-hand top corner where we talk about point of care, but with high analytical performance. There's another layer that adds on to this, which is that by making our near patient tests and our observations of a patient digital instead of human observations, which are non-digital, there is this opportunity for integration of data and having more and more computer-driven analytics, and that's a kind of unexpected potential benefit of future-looking point of care testing, which I'll come back to right at the end, both as an opportunity but also some difficulties. Um, so if we think about why we want point of care testing, I can tell you about the story of the kind of classic thing we're all very familiar with. The patient has a problem, talks to the clinician, if you're lucky, or phones somewhere. The clinician says you need a test, so you book a test. Then months later, when you actually have the test, you have to go to a clinic or a hospital. I've done this quite recently for a number of different things. You then go there, you have your blood taken, which is great. The blood will then end up somewhere in a lab. In the lab, sometimes it will get put on a machine eventually. The machine will run the test and provide a result to the lab. The result then has to be transmitted to where the clinician is. That doesn't mean that you get to see it. So the next thing is, of course, you phone up your GP and the receptionist says, oh, yes, your blood tests are in. Oh, look, this is what your result is. And if you're really lucky, you might get to see a clinician with the result at a later date. So this is the pain of centralized testing. And in theory, point of care testing is brilliant because when the patient presents with whatever the condition is, um, they do the test immediately and they immediately get the results. And so the patient and clinician can immediately do action. So this is why we think point of care is awesome. It's fast, portable, lots and lots of benefits to the health system. So that should be all great. And then the question is, how do we think that it will be possible? And I think that, the strongest analogy is that of microelectronics. We all walk around with powerful computers in our pockets nowadays in the shape of smartphones. And we can either make use of the components within smartphones. So digital cameras are now incredibly cheap because so many are manufactured to go in smartphones that we can then take our lab assays and make them readable by a smartphone camera. Um, Either in their phone handset or just use the electronics. So in the last 10 years, the cost of a digital camera has changed dramatically, and it's completely changed the way that we can build bioassay instruments. In parallel, what we can do is replace laboratory liquid handling with microfluidic systems, which are the fluidic equivalent of microelectronics. And so the theory is great. We can do better diagnostics. We can do point of care testing. And it's built on miniaturization of electronics and, in theory, miniaturization of the fluidics. So these are the three examples I'm going to give. We've got First of all, I'm going to talk, it's not to do with blood testing, but there's some parallels. I'm going to talk about how we measure the functional properties of bacterial cells directly in urine. Urine in some ways is easier to test than blood, but we can learn a lot from this. Then I'm going to move on to multiplex immunoassays, where we're going to measure, I'm going to show you how we can measure multiple biomarkers in blood. And then I'm going to talk about the future, which kind of combines these first two stories, and we start looking at cell measurement directly in whole blood, as is illustrated by this dip and test video here. So we have a platform technology. The simplest way of thinking about it is that we make very, very small test tubes. We're using this as a platform technology. It is a microfluidic system because we're measuring typically 1 microliter in capillaries that are about 200 microns or 300 microns in diameter. But there is a huge research field in microfluidics, and people make microfluidic devices using many, many different methods. And the way we do it is very, very distinct, which has some benefits and it has some drawbacks. And the most important thing about the microfluidic system we use is that it's genuinely mass produced. So we start off with plastic material. We feed it through a machine, which is just based in West Sussex, near Gatwick Airport. The machine produces reels and reels of plastic ribbon. And to give you an example, I think they've run this machine 3 times in the last 10 years. And every time they run it, they make 10,000 meters of material. And from 1 meter of material, you can cut it up to make 10 dip test strips. So it's very much analogous to the nitrocellulose paper that's within lateral flow tests. It's a mass-produced consumable. And the way we use it as test tubes is we cut short strips, and the strips all contain 10 tubes, and each tube is, I don't know, 20 millimeters long and roughly has a volume of roughly 1 microliter. So anything that you can do in a lab, in a test tube, you can then translate it to these low-cost, mass-produced materials. And because it's cheap and widely available, it has the potential to be a very powerful product. But at the moment, what's really cool is that we can use it in the lab to do lots and lots of cool experiments. And so we've done hundreds of thousands of microfluidic bioassays of various different flavors. And to just give you an illustration of the way that we can read out assays, so these readouts are all read using either a smartphone taking a photograph or using a smartphone camera. We've got color-based assays on the top left. So each of these is one strip of 10 1 microliter tubes, and we've got a yellow stroke red color-based assay. We've got blue and pink down here. We could look at whole blood directly. So these are whole blood flowing directly upwards in our capillaries. You can see these nice red lines. Each line is the flow of blood up a capillary. In the bottom left, we've got fluorescent assays, which is how we typically do immunoassays. And one of my favorite examples on the right, this looping video, is actually bacterial cells growing in the capillaries, and we're imaging them directly with a smartphone camera. And the only way we've illuminated them is to shine a white LED behind them in a particular way. And so you can actually see individual colonies growing. This is obviously a speeded up video. It's a time-lapse video taken over about 8 hours, and you can see individual bacterial colonies growing and count them, which is pretty cool. So the point about these visual optical readouts is that because we're doing an optical measurement, we can do lots and lots of the different bioassays. A surprising proportion of biological assays and even chemical assays that are done in laboratories by lab workers are actually color-based or fluorescence-based. And the basic paradigm is super simple. You start with a sample, you add some reagent, you get some kind of color change or fluorescence change, and really, since you're capturing it on a smartphone, the only thing we're really changing is that we're changing from a big glass test tube or a micro well plate down into a tiny microfluidic 1 microliter device. So my first challenge was point of care measurements, and the example I'm going to pick on first is to try and make measurements of bacterial cells directly in urine. And so the backstory here is that it's very important to measure the bacterial cells found in the urine of patients who have a bacterial infection, because when you have a bacterial infection, you have to be treated with an antibiotic, and it's incredibly important to do laboratory testing to check whether or not the bacteria are actually sensitive to an antibiotic when you're treating a patient with antibiotics. So the problem we have at the moment is that the way we test bacterial cells really is a historic method that's very similar to the origins of microbiology. So we tend to streak urine samples on a plate, a petri dish of agar. Agar was discovered in the laboratory of Robert Koch the late 1800s. Um, Although, of course, there was also pioneering work on lavender chip at this time because people used to grow bacteria on potatoes. Um, but no, on a serious note, we also have methods for measuring whether or not bacteria are sensitive to antibiotics. Really going back to the origin of antibiotics, we typically spread bacteria on a petri dish, and then we put some antibiotic in a paper disc, and then we can measure whether the antibiotics kill the bacteria. So it's a functional cell assay. You're growing the bacteria and seeing their response to antibiotics. And then in the 1980s, this became replaced in some cases by micro well plates. So we work in 100 microliter volume and we can measure antibiotic sensitivity. So we feel it's a natural progression, and it is now about time that we moved on from microplates which are as old as I am. And instead of using a micro well plate with 100 microliter volumes, we use these tiny little tests. You can see how big they are held between the fingers. And if we compare here a microplate method, obviously this is a scaled image with the capillaries inside our test strip, you can see you get identical bacterial growth with 100 times smaller volume. And so this tiny little strip, which is read by a smartphone camera, allows you to shrink your assay and then take it into a point of care setting and do a test very rapidly near a patient. The really crucial thing for us is that what is hidden in this historic thing, so you can complain all you like about how old the microbiology methods are, but one really important thing is we have decades of experience of using these tests. So if you come up with a new technology, one of the things you really need to do is to prove that your assay is the same as the standard method. So what we've been doing a lot for the last 5 years is doing very, very standard antibiotic sensitivity tests. But the one difference we're making is instead of doing it in a microwell plate, we're doing it in our 1 microliter dip and test strips. And the way we do that at scale is we actually integrate our microfluidic system with microplates, which allows us to do very large scale experiments. And so in an afternoon, we can set up an experiment where we image, in this case, 12 by 12 times 4 different strips. So each of these is one of our test strips, and each test strip has 10 different conditions in it. So we can then film this by time-lapse photography, and we can get bacterial growth analysis in a time-resolved setting. And if we sort of imagine individual capillaries along here with a time axis going downwards, what you can see is bacterial growth where the color in the capillary is changing from blue when there's no growth to pink when there is growth. What you can see is in each individual capillary, you can measure the kinetics of bacterial growth. And for example, this strip here has bacteria which are all killed or inhibited by an antibiotic. And this strip here, the bacteria can grow in the presence of antibiotics. So you can detect the difference between sensitive and resistant organisms in great detail. So because we have a low cost material, we can make thousands of our devices. We've been able to ask all of these really, really important questions about what happens if you miniaturize a standard microplate assay, and even better, what happens when you directly test the urine and don't bother with the overnight petri dish plating, which is the current reference standard method. So we can worry about whether the urine interferes with the assay. We can look at whether or not the bacterial cell density in the urine affects our assay, and we can do lots and lots of cool things about this. And most importantly is we can optimize fully and then check the accuracy of our assay, and we can work out how to get smaller antibiotic susceptibility tests to actually work with accurate results. So we can find out all sorts of interesting things. So for example, if you take your urine sample and dilute it tenfold, it slows down the growth detection by an hour. So if we want to have a rapid test near a patient, that's great, because what that says is that we should dilute the sample less and we should have higher starting cell concentrations. So a very quick plot here shows that as you have higher bacterial densities, it takes less time to detect. But if we trade that off, we need to be aware that if we put higher amounts of bacteria, again on the x-axis down at the bottom, what you can see is that what happens is if you put too many bacteria in your assay, you start to get lower accuracy and you get false resistance, which is something that's well known, but we can document this in great detail for our microfluidic system. So what that means is that you have to make a decision about whether you want a fast result or an accurate result. Um, because the faster results are likely to give more and more errors in your readout. And so what that allows us to do is to do a kind of optimization matrix where we look at all of the different factors that affect accuracy of our assay, and we can layer over the top of this something which I think is really, really crucial with any diagnostic development program, which is to think about the actual use case and the setting, think about the clinical pathway, think about how this health system operates, because we can now ask very specific questions in a healthcare setting and say, well, do you want a result with this accuracy after this many hours, or would you prefer this accuracy after that many hours? And you can actually look at the consequences for patients for having a faster, less accurate assay versus a slightly longer time wait, but a more precise readout. So having this system has been incredibly powerful. And the most important thing I would emphasize is that in order to come up with this, we had to do an awful lot of experiments with our microfluidic system, which was possible for us because our system is cheap and mass produced. But this kind of development is urgently needed for all of the microfluidic systems out there. I would say as a general comment, there's a lot of proof of concept work done showing that people could do assays in clever ways, but it's very, very important that we pursue the optimization and systematic analysis as well. So we've got plenty of evidence now to say that we can definitely miniaturize antibiotic susceptibility tests, which is in agreement with other people around the world with different technologies. And we're pretty confident now that we can we can test urine directly, and that allows us the potential for doing portable urine antibiotic susceptibility tests, and we can worry about whether that's actually what the health system needs. We have to think about what's actually useful in the real world. Whether or not the point of care system is superior is a subject of a completely different seminar. But if you're interested in that discussion, I'm very, very interested in that kind of thing. So where we are at the moment with this point of care urine testing sample is we're running a product development award funded by the NIHR, which has been really, really powerful. We're into the third year now, and we've got a much larger database of real sample testing. We're well over 350 samples. We've got a target to test 600 samples within that study, so we're well on track to do that. But alongside that, absolutely crucial is that we've got insights into what the health system needs. So, as a conclusion, point of care testing is technically possible, and it's possible through miniaturization. So if we move on to the next thing, so one of the things we want is we want a near patient test, but the other thing that we need to do is we need to move away from single test, single question, traditional diagnostic testing, where you look at the patient, you come up with a conclusion, you say, I want to test this one thing, and then based on the test result, you make a decision. And it's very, very clear that in the near future, everything will be improved by data-driven diagnostic testing. So my little picture here shows how we currently do medicine, which is, it's a very human process. And of course, everything needs to go towards computers because that's the future. And we need to replace subjective judgments, which are pattern recognition, with objective data-driven analytics. And this will have a massive effect on the way we interpret test results. And one of the things that we need to be able to do, I think, to do that is to be able to quantify panels of biomarkers. And what I mean by that is that we've got to make more than one measurement in one test. And ideally, what we want to do is what I would term multiplexing, which is where you take a blood sample or another patient sample and you measure a panel of um, different parameters in a single go. So you can go from a single blood sample into, if you like, a barcode perhaps, or a fingerprint. Um, so the idea here is that our vision would be to take the same kind of core technology platform, which is microfluidic, try and maintain the simplicity of lateral flow, and use our readout as the smartphone camera, and then make measurements of ideally 10 or more targets. And we want to get quantitative measurements, not detection. So we don't want a positive or negative. We want to have quantitation, and that allows us to come up with data that can feed into an algorithm, and you can get diagnostic fingerprints. So then the question is, what do we want to measure? And I think it's worth just remembering blood is complicated and it's hard to measure things in. There's a load of different things. I'm not going to go into detail with this slide, but just to highlight, there's lots of different things we currently measure and lots of things we want to measure in the future in blood. They're all present in different concentrations, and there's all sorts of other things. And the other thing I wanted to emphasize very, very quickly is that quantifying biomarkers in blood is difficult because there's a lot of stuff in blood. So you have very low concentrations of some biomarkers, very high concentrations of other ones. Nearly half of blood is actually cells, not liquid, not plasma. The cells are very complex and dynamic and changing all the time. The concentration of protein in blood is very high. And the other thing about blood is that it varies a lot. Within an individual, it changes all the time. The concentrations go up and down. Probably your hydration status affects the concentration of some blood biomarkers. So it's not an easy thing to do. But one of the most powerful platform technologies that we have is to use antibodies to make measurements. We call these immunoassays. Again, apologies to anyone in the audience who are very familiar with this kind of technology. But very simply, the way that an immunoassay works in any platform and the way it works within our capillary film. So if you imagine this is one capillary here, is that we coat the inside of the capillary with an antibody, and the antibody molecule has exquisite ability to bind to a single target with very high affinity. If you then flow blood through, the biomarker in question will get captured by the antibody, and hopefully most of it, or a set proportion of it, will end up bound to the wall. We can then flow another reagent through, which is a second antibody which binds another part of the antigen, which again improves the specificity of the test, because only the target antigen will be measured. And that antibody is tagged with a little protein, which is a very powerful enzyme, which then converts a substrate from one color to another. It might be a colorimetric substrate like a yellow color, or it might be a fluorometric fluorescent dye. And the great thing about immunoassays is you can really detect very, very low concentrations of target molecules. So the enzyme amplification gives you fantastic sensitivity. This also requires a multi-step process. So enzyme immunoassays in a laboratory, anybody who's worked like me as an immunologist will have done lots of these assays. One of the things you spend a lot of time doing is washing. So the sensitivity is great, but it does come at a cost, which is that you have to have efficient washing and you have to do lots and lots of sequential steps. But the great thing about our test strips is that you can have different antibodies measuring different targets in each strip. And so we've packaged our test strips into little plastic holders, which allow us to process multiple reagents and samples through. And they look a little bit like this. So on the left, we have a lab-based system, which fits into a little case. We also have a near-patient field testing system, which is not much bigger than, well, it's about the size of a sort of handheld carousel. We need to have a few little liquids that go into this, so it's not an entirely dip and walk away test. And we can do immunoassays in these capillaries. And in fact, going back quite a long time, back in more than 10 years ago, we set up a business to do this called Capillary Film Technology. And this business has been driving towards commercial application of immunoassays in microcapillary film. And we actually developed, using some brilliant SBRI funding, an instrument. here, which is fully automating the process of doing immunoassay in our capillary strips. So this prototype instrument can perform an immunoassay from a blood sample in capillary film. But our favorite way of doing the immunoassay works like this, using a rather nifty trick of a fluidic siphon, which allows us to take a drop of liquid, which could be the blood sample or the reagent, and flow it through gravity alone. And I've got a little video of this. What this allows us to do, the most important thing is if you imagine those red strips are blood, in a moment, the blood will be fully washed away by a wash buffer. You can see the red lines are disappearing. And then in order to add our next reagent, which would be the detection reagent, we can simply pick it up and move it to the next step, or we can put it in a carousel. And this can then put, you can see there's a blue dye in there just to show that the next liquid has gone in. So with very, very minimal reagents and manipulation, we can do these complex multi-step immunoassays, and we can measure panels of 10 different biomarkers in one little plastic strip. So where we are in terms of validating this platform is that we recently published a study that was done in a field setting, in the sense that it was done in a hospital in Thailand. We were looking at detection of dengue virus, which is a very important pathogen that everybody in any kind of tropical country has heard of, because it affects more than half the world's population. And we were able to simultaneously measure 4 different targets, each of them measured in duplicate, plus some positive and negative controls in our test strips. Essentially, what we found is that doing an immunoassay in our tiny capillaries is much faster than a plate ELISA, but it gives you very similar results in patient samples. But one thing I also wanted to highlight, which is really important, is that we do not have a product, and we don't have a clear pathway towards a high value product. And I, again, it's probably a whole nother seminar to talk about barriers to commercial development of diagnostic technology. But it's cool technology, and it shows that you can make measurements of multiple targets in one sample, and we can quantify the target, the biomarker target, using immunoassay in a microfluidic system, and it is compatible with a near patient, low cost, portable setup. So it's technologically possible. So then, final example I wanted to give you is one for the future. We have worked closely with platelet and blood researchers at the University of Reading, particularly Chris Jones, to explore this idea that we can simply dip our test strips in, and when we dip the test strips in, the blood flows up by capillary action. But if we stimulate to cause coagulation or platelet activation, we can really dramatically affect the flowability of blood really quite fast. And because the blood is flowing in a capillary, we can then quantify the flow. And the idea is we're kind of looking at what we learned from testing bacteria in urine and applying it to whole blood. And what we're able to do with our platform is to do multiplexing, near patient, and cell function assays. And just to throw in an extra thing, it's not just about platelets. We can also measure other cells which are present in blood. This is a very preliminary proof of concept that we could potentially measure T lymphocytes in whole blood. So the crucial thing is that we've got to think about what we're trying to do. We are not trying to study the behavior of blood. What we're trying to do is to measure the processes which are triggered when blood is activation, which can be platelet activation, coagulation, and also we get quantitative kinetic information, which tells you about how fast the blood responds to stimulation. What we're not trying to do is to actually study platelet function. That's better done in other things. What we're trying to do is come up with something that's super cheap, potential for testing regularly and rapidly. It's got to be multiplex, and it's got to have a readout which doesn't require laboratories. So the visualization happens through smartphone camera technology, and it becomes even more important to do platelet function testing and blood function testing near the patient, because if you draw blood from a patient and then leave it for a few hours, it changes and it may well not give you correct results. So we need to be able to test people very near the patient with very fresh blood. So this is an example in the future, and I kind of trying to build a paradigm where we have really low cost cell function measurements, where we start off with a blood sample. We may need to do some processing. We may need to filter and separate, which is something that we can do using existing technology. We then stimulate. Most importantly, we stimulate with a panel. So we're making this a multi-parameter measurement, and then we can read out some kind of parameter, which can be done using the digital camera platform. So if you put these together, you can then make multiple measurements in blood, and that's the kind of where I see a major kind of 5 to 10 year research project. So overall, all of these benefits from the same core technology, and the most important thing is that it's low cost, which means we can do lots of experiments, as I said. So picking these three examples and this core technology, I think it's true to say that we, and of course many other people, have got all sorts of other very cool microfluidic technologies. If you ever want to find out more about these, I would encourage you to go to a microfluidics research event where you can go and be bewildered with every possible shape, size, and material you could imagine for making measurements in tiny, tiny things. And that's really important to understand that the technology is around. With our own setup, we can do immunoassays, we can do bacterial detection with or without labeling. Another really cool example I like here is that we can measure not just bacteria, but we can measure viruses. So we can measure bacteriophage, which kill bacteria, and you can actually see the killing of the bacteria in a bacteriophage plaque assay. We can do absolute cell counting, all sorts of different things. So we can use that for all sorts of things. But I think one thing I have learned um, alongside that is that one of the big difficulties with diagnostics innovation is that there's lots of things that you need to deal with. And as well as getting the technology to work and the biomedical science to work, it's important to step back and say, do you know what? Actually, the way that you use testing is very important. And even something as simple as the instructions that go alongside the test are really, really important too. And the technology on its own can't change anything. What we need to do is have an innovation mindset. And an innovation mindset is really, really vital. And if we look at the evidence from the field, if we actually ask how long it takes for new diagnostics technology to be widely adopted, I don't know where this statistic comes from, but I've heard it from a number of different places, that if you actually look and see, it can take more than a decade for a new technology to be widely used by the NHS. What we need to do, therefore, and there's lots and lots of infrastructure out there that you can work with to look at this, is to plan into the future and make sure when you're developing new technology, you think about who's going to use it and why. And what I've learned is a very powerful tool. And this is developed by my collaborator and colleague, Julie Hart, is what she describes as a lean mixed methods broad stakeholder approach, which is kind of obvious when you look at it, but it's very powerful. Essentially, what it does is it looks at all the people involved in the diagnostics pathway and maps out what will be the barriers you have to overcome. It also identifies health economic benefits and health economic barriers to adoption, and it emphasizes the importance of mapping where your diagnostic product could change clinical practice. And this allows you to define your product requirements in a way that means that if you could develop a product and launch it, you maximize the chance that the product does what the customers and the end users and the patients actually need. Um, the other thing I've learned and seen a lot is there's a huge barrier of real world evaluation, and you need to be able to prove in the real world with a field trial that your diagnostic test is actually beneficial. So what that means is, if we zoom out a bit and don't get sucked into just really focusing on the technology, there's a lot of simultaneously challenges which need to be overcome if we're going to come up with this new near patient multiplex data driven next generation of diagnostic tests, because you've got to worry about what the biomarkers are, you've got to get a good device and instrument, you need a commercial plan, you need the clinical partnerships as well, you need to make sure there's patient benefit and that you've got patient and public involvement. All of this costs money. There's a lot that needs to happen. And I just wanted to add one additional thing that can be a bit of a challenge, which is that I think it's recognized, and this Future Blood Testing Network has had great presentations identifying this, that it can really grow in cost as you develop products closer to market. That actually doing a concept experiment might be a PhD student to prove that something's possible, but by the time you actually get to product launch, you really need to be able to raise somewhere in the order of 5 to 100 million pounds, and that's getting to the point of an IVD being regulatory approved and being available for sale. And I have no idea how much it then costs to drive widespread uptake of your product, but it's going to carry on costing money. And this has to be balanced with the value of a diagnostic product. And you can do very simple back of the envelope calculations to say that if you can come up with a low cost diagnostic, it's probably got to cost 10, ideally, possibly as much as £100. And you really need to think about how many tests you could sell in a year or how many tests would be used by a health system. And what I keep coming back to is that on a very, very rough scale, log scale, ideally you need to find opportunities where your technology or product could be used 1 million times a year, because otherwise it's very difficult to raise enough money, if you go back one, to actually develop a product and launch it. So there's a lot of commercial worries or difficulties coming back to this kind of raw sum. But anyway, it's definitely worth looking at as well as the core technology. Um, so one final thing I wanted to raise, which I think is really interesting, something I'm looking at more closely now I've moved into a department full of health data scientists, is that implementing data science principles is inevitably going to have an effect on the way we do testing. Because in the past, I've said this already, we have had this paradigm for diagnostic testing that you have a question, you have a patient, you do a test, you get a result, and that's interpreted in the context of wider clinical data. We're moving away from that to much more complex interpretation where test results may have an unexpected input onto some kind of algorithm. And I think I can't see how this isn't going to affect the way we develop diagnostic test products. And one way of thinking about that, which is a little bit abstract, but it helps me to understand it, is that if you look at the way that diagnostic tests are regulatory approved, it comes from a background of analytical science, where the regulations require you to define your product very much as an analytical chemistry tool. I'm going to give you a couple of examples which I think are helpful. But when you're developing a product, you need to follow a pathway. But the thing is that's interesting is that the diagnostic test development pathway, whether that's the regulations or the way that we develop medical devices through a product development pathway, is really derived over decades of experience developing old style diagnostic products, and it's not been developed with data science as the readout in the end. So if I pick an example, so this is a kind of classic threshold diagnostic. This is actually cardiac troponin distribution. So you can see patients in red have had a heart attack and they have a higher level of cardiac troponin, and you can stick in a threshold value, and you can use very, very classical analytical science to quantify the performance. And you can write a specification which has got all the things you'd expect to see, coefficient of variance at different concentrations. It's very much a single measurement technology. And this is how a specification of a product would look, and this is what the regulations are expecting you to have. And then you can measure your performance using very conventional statistics and approaches. But what we all want to see is more sophisticated interpretation of results. So, for example, if we take the same kind of testing system, maybe not cardiac troponin, but another blood biomarker, if we actually monitor individual patients, what we know is that some of the variation, if you imagine this is the variation in the population, again, if this isn't very clear, this is sort of developing ideas, it'd be great to get feedback on whether this is understandable. Individual patients may have much more stable levels of a biomarker. So if you measure them multiple times, you can work out what the trend is for the individual. What this allows you to do is potentially identify abnormal events because of a small change in a biomarker concentration. And in the context of the whole population, you would never be able to score this as an abnormal result, but for the individual you can. And the other thing, which I think has been a well-trodden path in things like blood pressure monitoring, is that you can also detect through monitoring perturbations. So when you end up with a higher variation, so actually the population of data points changes even if the value doesn't go outside normal range. The question for me, though, is that if you go back to IVDR expectations and the standards that are out there to help you develop products, you end up having to sort of redevelop the way that you define your product. So I think that's something that's going to be a big deal. Another thing that feeds back is that people working in the data science worry about things like instrument drift. In the situation with instrument drift, the idea is that you actually get the reverse situation, which is that you have a stable measurement in a stable parameter in a patient, but perhaps you buy a new cartridge or there's a calibration change or something happens to the instrument you're using to measure the patient. You may then end up with a shift in values, and the shift in values may trigger an incorrect interpretation of the data. And again, I have no idea how that affects the way that we define IVDs when we're developing them. So I think we need to work together as a community and think about these bigger problems that come from a data-driven approach. So hopefully some of you are interested in that. I just wanted to end with thanks to the team. So as I mentioned at the beginning, I'm now at the University of Southampton. I've been at the Reading School of Pharmacy for a really long time, and I'm really delighted to say that Dr. Sarah Needs has taken over. She was a postdoc working with me for more than 5 years, and now she's leading that lab. So that Reading Research Group is continuing, and I work very closely with them. I also mentioned Julie Hart, who has been fantastic as a guru of diagnostics innovation for me. So a lot of what I've seen about that, I've learned from Julie. And I worked for a long time on microcapillary technology with Nuno Reis and the late Patrick Hester, who's been fantastic in supporting it. And also, we couldn't have done any of this work with funders. So thank you all for joining the seminar, and that is the end.